Hello, everybody. Welcome to this Gen webinar. It's entitled High Throughput Synthetic Biology, Synthesizing Designer Yeast Through Automation and Acoustic Liquid Handling. Synthetic biology is a relatively new field, and it's already drawing huge amounts of attention in the life science community. This technique focuses on producing engineered organisms to perform new and useful functions. It aims to develop technologies, methods, and biological components that will ultimately make the engineering of biology safer and more reliable. Today's Gen webinar has been designed to illustrate how a specific approach using acoustic liquid handling complements synthetic biology research and provides experimental data from streamlined workflows that show increases to overall productivity. I'm John Sterling, Editor-in-Chief of Gen, and I'm going to serve as the moderator. And we've assembled a panel to discuss examples of the application of acoustic liquid handling to synthetic biology. So let's meet them. Randy Dyer, Senior Product Marketing Manager at LabSite, will introduce concepts underlying advanced acoustic liquid handling and how they are applicable toward many aspects of laboratory work. He'll also demonstrate how LabSite's Echo 500 series liquid handler can aid in streamlining production workflows. Dr. Patrick Yidzi-Kai is co-director of the Edinburgh Genome Foundry at the University of Edinburgh. Patrick will discuss his work on the synthesis of artificial yeast chromosomes. He will also describe how acoustic liquid handling technology has aided his research by significantly increasing the speed at which he can genotype synthetic products and downscale the volume of DNA assembly dramatically. After the panelists make their presentations, there will be a question and answer segment. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible. So if everyone's ready, let's get going. Randy Dyer will be our lead-off presenter. Randy?
Thanks for the introduction, John, and thanks to everyone who has joined us. I want to start off with an overview of LabSite and what our products have done for researchers across many areas of life science. LabSite invented a technology that uses sound energy to transfer liquids. Then in 2003, launched the first product using this technology, the Echo Liquid Handler. Today, Echo Liquid Handlers are used globally by major pharmaceutical companies as well as smaller research laboratories. Echo Liquid Handlers have offered users a number of advantages over traditional methods in a number of areas, including pharmaceutical development, cancer research, and diagnostics. Over the years, LabSite has grown to additionally manufacture and sell consumables, automated robotic systems, and software as complementary components of what we consider to be end-to-end solutions for a number of sample preparation workflows. Here's a snapshot of what our users have achieved in genomic application. For each application, you can see that use of the Echo Liquid Handler has dramatically cut costs to produce a reaction. These costs are estimates based on information that we pulled from various publications, and the dollars may not be exact, but that shouldn't take away from the level of miniaturization our users have achieved. Also, keep in mind that using acoustic liquid handling eliminates the use of tips, which in turn generates less waste, avoids cross-contamination and carryover errors, and increases throughput. Also, low-volume transfers can be used to create higher density pools. Later in the webinar, Patrick is going to go into the details of what he's been able to do with the Echo Liquid Handler, and you'll again see that the level of miniaturization you can achieve with the Echo is enabling for any laboratory trying to scale their workflows. I'm going to go through the next few slides just to provide more detail around our patented technology and how we incorporate it into our offerings. LabSite's flagship product is the Echo Liquid Handler. We recommend different models based on the volumes, fluid types, and throughputs required for your application. Every system arrives in the lab pre-programmed for a range of fluid types, so you don't have to go through any optimization process when you want to transfer different fluids. Also, every system has the ability to transfer fluids from any well to any well and any number of wells. This offers several benefits when you're cherry-picking samples, replicating samples, titrating samples in a concentration curve, or creating complex patterns for assay development or arraying. Echo liquid handlers use an acoustic transducer located under the source microplate to emit acoustic energy through the bottom of the plate up to the meniscus of the sample that you want to transfer. When the acoustic pulse hits the meniscus, a droplet of the sample is ejected upward into the destination well or onto some other surface. You can use the echo to transfer to microarrays or chips, a variety of destination vessels. Depending on the model, the droplet volume can be 2.5 or 25 nanoliters, and the rate of droplet ejection can range from 200 drops per second to 500 drops per second. And that's how we build large volumes, by rapidly transferring these highly precise and reproducible droplets. Using a proprietary technique called dynamic fluid analysis, echo systems can acoustically sense changes in fluid properties and adjust in milliseconds prior to transfer. This helps us maintain transfer performance despite hydration, evaporation, or changes in the height or shape of a sample's meniscus. Also, dynamic fluid analysis happens automatically as samples are transferred. As I said, it happens within milliseconds, so you don't notice it, and it ensures that every sample is transferred precisely and accurately as you go from sample to sample. And remember, all of this happens without any contact with the fluid. That cuts down money spent on tips or time spent repeating experiments because of pipetting errors. This slide highlights several unique advantages of the echo's liquid handling technology. It takes just a second for the echo to position its transducer under any well of a source plate to transfer a sample or reagent. And the samples and reagents can be transferred in any order. This allows you to assemble components of a reaction many different ways. You can do this to evaluate the impact of changing concentrations or using different components. This is ideal for assay optimization, compound screening, or pooling libraries. Now, without tips or tips changes, the echo is transferring these individual components very quickly. Next, I want to talk more about our products that complement the echo. We offer a suite of software applications that vary in complexity and intended use. You can choose to define every transfer graphically or with an Excel file. You can use the wizard to set up dose response assays or create custom transfer patterns for chips and arrays. We also have an application that will allow you to analyze and compare samples in your library over time, and look at changes in viscosity or hydration, and you can use this for quality control of your sample library. All of our software applications use a number of wizards and graphical tools to help users quickly set up their protocols and visually see what's going to happen on their echo liquid handler. Also, we offer a robotic system, the Access Workstation. The Access readily integrates the echo liquid handler with a variety of devices to build fully automated systems. Every Access Workstation comes with Tempo software. The Tempo software greatly simplifies the conversion of an echo-only protocol created using one of our echo software apps that I just mentioned into a fully automated routine. Users simply import their echo application that they want to use, and then add steps like spinning or sealing or peeling, and save that protocol. And behind the scenes, Tempo software develops a very complex and dynamic robotic schedule automatically. So for users, we're trying to make the utility of the system very, very easy and very accessible. And by combining our Echo liquid handlers with targeted software applications and integrated robotic systems, we've developed fully automated systems that target end-to-end sample preparation workflows in a number of applications. We have demonstrated our utility with application notes and publications from our users in genomics, also in biochemical analysis, and as well as drug discovery. Essentially across the life science space, our users have seen benefits of working with LabSite. And before I let Patrick tell you about his experience with the Echo liquid handler, I want to go back to one slide. This is a great statement of what our products can accomplish in a handful of applications. But I also want to point out that our users use the Echo in many applications not covered here and see the same benefits. It's truly an enabling, revolutionary technology. And with that, I'll turn it back to John.
Thank you, Randy, for that overview of the ecosystem and how it can play a key role in streamlining production workflows. We very much appreciate the application focus of your presentation that you demonstrated, so thank you very much. If you're just now joining our webinar, a big welcome. Just a reminder that there will be a question and answer segment after the last presentation. Please submit your question into the box on the left-hand side of your console and then hit submit. Our next speaker is Dr. Patrick Yidzi-Kai, and he's ready to go. Patrick?
Thank you, John, for the kind introduction. It's great to be here to talk about the research project of synthesizing yeast genome from scratch using automation acoustic dispensers. So in the past 20 years, we have made tremendous progress in reading out the DNA encoded in the organisms. So right now, we are trying to write DNA, and the progress has not been as fast as we want. So today, we would like to develop technology we can write DNA very efficient, so we can go through the reading to writing loop as fast as possible. So this slide shows you the progress of writing DNA. So starting from 1979, we can write a small piece of DNA and publish a Nature Science paper, but the competition goes up really fast. So right now, the milestone is set by Craig Venter Institute by resynthesize a bacterial genome in 2010. And that's about 1 million base pair. So we have gone a long way since 1979. And today I would like to tell you a little bit about the project we are undertaking as a consortium to synthesize the synthetic yeast genome. So this is not just a lot more DNA, so it's about 12 million base pair, but this is also the first eukaryote genome to be redesigned and resynthesized. So to start with, I would like to introduce you to the budding yeast, Saccharomyces cerevisiae, a fantastic organism to work with. So what's really in the name? So Saccharomyces means sugar fungus, cerevisiae means found in beer. So that tells you all about these organisms. So about 5,000 years ago, 3000 BC, we know how to use yeast to fermentate alcoholic beverage. So in the left corner, some ancient Egyptians are using some long straw to drink alcoholic drink. This is one of my favorite quotes by Benjamin Franklin. Beer is the living proof that God loves us and wants us to be happy, and I think that's largely true today across the entire world. So I would like to tell you a little bit about the relationship between the yeast and the human genome. So yeast is one of the simplest eukaryote genome. So yeast has 16 chromosomes and human has 46 chromosomes. Yeast has 14 million base pair of DNA and human has 6 billion base pair of DNA. And yeast has 6,000 genes compared with 20,000 genes of a human. And it's important, we're going to get back to the point of 6,000 genes in the yeast. Yeast is single-cell organism. That means one cell is one yeast. And human has 50 trillion cells. Yeast can be haploid or diploid. It can be mate A or alpha, just like human can be male or female. So before we redesign this organism, as engineers, we would like to set up some design principles. So the first design principle is do not harm, even to the yeast. So we would like to maintain the fitness as close as to wild type. And second is we would like to stabilize the genome. So this is similar to the first design principle. We don't want to harm the yeast. We want to have a very stable genome after we resynthesize it. Otherwise, nobody can use it for industrial purpose or research purpose. And the third is try to increase the genetic flexibility. We want to make it as flexible as possible. So it sounds like we have two competing agenda here. On one hand, you want to stabilize the genome, but on the other hand, you want to stabilize the genome to increase the genetic flexibility. And I will tell you how we achieve both goals in the next few slides. So these slides summarize what we do to the yeast genome in terms of design. So the goal here is to design, to construct, and to integrate entire sensitive version of the 12 million base pair Saccharomyces cerevisiae genome. In the last few slides, I told you there are 40 million base pair in the wild type genome, and now I'm saying 12 million. That's because we're going to trim down 2 million base pair from the wild type genome. So we delete a lot of elements which we don't think are necessary for the viability of yeast. For instance, repetitive elements, transposons, introns. So for instance, we don't think alternative splicing is essential for the viability of yeast. And we also delete all the tRNA genes. But as you know, tRNA genes are actually essential for the viability because it's low in translation. But because tRNA genes are the genome damage hotspot, which I'm going to expand in the next few slides, so we remove all the tRNA gene from the 16 chromosome, but we create a 17 chromosome we call tRNA neochromosome to relocate all the tRNA genes in one chromosome. So the idea of deletion and relocation is try to study the essential role of these elements in the genome. And this is also another principle of synthetic biology is learning by doing. And the goal of deleting some of these elements, such as tRNA genes and transposons, is to fit the design principle of stabilizing the genome. Second, we modify the DNA to some extent. So we embed some watermark sequence in the coding region called PCR tag. So these are the watermark we put into the gene, which would translate to the same amino acid, but they're very different in DNA level. And we also change all the stop codons, TAG codon to TA. So the idea of embedding the PCR tag is to quickly distinguish wild type and sensitive sequence. And the idea of swapping all the stop codons TAG to TA is we want to bring the 21st amino acid as a safety measurement for sensitive yeast. And finally, I think most exciting thing is we put loss psi everywhere in the genome. So we put a loss psi downstream of every single gene. So at the end of the day, we have a genome carrying over 6,000 loss psi's. So with this system, we can study the genome structure. I'm going to expand on each point in the next few slides. So as I mentioned, we delete and relocate a lot of elements. So these are removed by design. So for instance, we remove all the transposons. So these are the jumping elements in the genome that cause genome instability. And also we remove the introns to study whether alternative splicing is essential or not for the yeast genome. And most exciting design we have done to the yeast genome is we build what we call scramble system, which we can on demand turn on the genome rearrangement to study the genome evolution, genome structure, and organization. And also we can use it to minimize the genome, and with the hope to identify minimal genome sets. So when I said minimal genome sets, I mean we believe there's not just one minimal genome. There must be many minimal genomes, because when you delete one pathway, the other pathway becomes essential. Okay. So we have two competing agendas here. On one hand, we want to develop a super slick designer genome, but on the other hand, we want to be able to expand the genome by scrambling the genome. Because we don't want to just make one genome, we want to make billions of genomes at the same time. So we made some modification to the DNA. So we bring in what we call PCR tag, which encodes silent mutation in the genes. So we can quickly use PCR to identify whether a piece of DNA is synthetic or wild type. We also swap the most rare codon, TAG codon, in the genome to become TAA. So at the end of the day, there will be no TAG codon. Then we can reassign the 21st amino acid to the genome. So this would be a safety measurement for the sensitive yeast, because once the sensitive yeast escapes from the laboratory, it's entirely different vocabulary from the wild type, so it cannot crossbreed with the wild type yeast. So this slide shows you what's the design of a PCR tag. So PCR tag is really short DNA fragment. It's about 25 base pair, and each PCR tag is about 200 base pair to 500 base pair apart from each other. They're at least 1/3 different from the wild type DNA. So if you look at the two PCR tags I'm showing in the bottom of this slide, they encode the same amino acid, but from the DNA label, they're at least 1/3 different. So with a pair of DNA, a pair of PCR tags, we would be able to quickly identify a piece of DNA is wild type or synthetic. This is really important for us to quickly genotyping the entire chromosome. So in order to quickly rearrange the genome, we built in a system called Scramble by putting in a loss-P same site downstream to every 3' UTR in the genome. So remember, we have about 6,000 genes in the genome. So at the end of the day, we build in about 6,000 loss-P same sites. So loss-P same site is 34 base pair symmetric DNA. It's a special kind of loss-P sequence because it's symmetric. So when you flank a piece of DNA with a pair of loss-P same sites and you express a Cree protein, you got 50% chance of deletion and you got 50 chance of inversion. And the combination of the two also give you insertion. So once you express a shot of Cree, every two loss-P out of 6,000 loss-P can recombine into these three configurations. So with this design, we would be able to discover genome structure rules. We would be able to quickly evolve the strain towards different phenotypes. One of the goals of the Synthetic Yeast Genome Project is to stabilize the genome. So by deleting a lot of repetitive elements, and also we remove all the tRNA genes. So there are 275 tRNA genes in the yeast genome across 16 chromosomes. And the tRNA genes are genome damage hotspot because they are the insertion hotspot for transposons, and also they're being transcribed really heavily, and it causes collision of the polymerase. So by developing a near chromosome, which is the additional chromosome, we put all the tRNA genes in one location. We call it party chromosome for tRNA genes. So this slide shows you the design footprint for chromosome 9R. So this is the right arm of chromosome 9. So this is the shortest chromosome arm in the yeast genome. It's about 100,000 base pair. So what we are showing you here is the genes are encoded in arrows. So the red arrows are essential genes. So as you can see in this particular chromosome arm, we only have a handful of essential genes. And the light blue arrows are non-essential genes, meaning when you delete them in a single mutant context, the yeast can still survive. And I should have mentioned in the yeast genome, there are only 1,000 essential genes and 5,000 non-essential genes. But this is in the single mutant context. The green bars in each arrows are the PCR tag we embed. Again, these are watermark sequence across the entire genome. And the green diamonds are the lost psi. So we put lost psi everywhere in this chromosome. And it's about 43 lost psi in this particular chromosome arm. So we designed this in a computer program called BioStudio, and the design was done automatically. So we redesigned this 100,000 base pair, and it was synthesized by a company called Codon Device. They synthesize this DNA as a circular bag or a yak. So we transfer this bag, which was tagged with a marker called Leu2, into a Y-type diploid. So the Y-type diploid has both copy of the Y-type chromosome 9R. When you transfer this back, then you increase the copy number of the right arm of chromosome 9 into three copies. And then we truncate one of the chromosome Y-type chromosome arm and target with ULA3. So at this point, you got a cell which has two copies of chromosome right arm, Y synthetic, the other is Y-type. And then you sporulate this diploid and look for Leu plus, ULA plus cells. So on the right-hand side, this is the example of how we use PCR tag to identify a piece of DNA synthetic Y-type. So when you run Y-type PCR tag against Y-type genome, you only got the amplicons of the Y-type DNA. And similarly, when you run the synthetic tag against the synthetic genome, you get the amplicons. So we need to use the PCR tag to quickly identify whether a chromosome is entirely synthetic or not. So after we finish synthesizing the chromosome 9R, we need to categorize the phenotype to see where it actually fits our design principles. So what we are showing here is three strains. The first strain is the Y-type yeast, and the second and the third are two independent isolates of the synthetic yeast, which are completely powered by the 100 kb chromosome 9R. We look at different conditions of different media of different temperature, and we could not see any difference in terms of colony size. And colony size is a very sensitive measurement for the fitness of the yeast. So this actually fit really well of our first design principle. The synthetic yeast is very close to the Y-type. Now we have a synthetic yeast we can play around, and we want to scramble the genome. We want to be able to tightly control how much scramble we like, because as soon as we start scrambling the genome, we start losing the cell, because once we delete one essential gene, the cell will be dead. So we need to have really tight regulation of the Cre protein. So this is a special kind of query construct we have. So it has two kinds of regulation. The first kind is transcriptional. So this particular query construct is under the control of a promoter called PSCW11. So this is a doctor cell specific promoter. It only turns on once and only once in the lifetime of a cell when the cell is a doctor cell. The second level of regulation is post-translational. So we hook up the query with estrogen binding domain. So when there's no estradiol, a small molecule in the cell, this query protein is bound by HSP90 chaperone in the cytoplasma. And then you add a little bit of estradiol, it changes the conformation like the HSP90, and then the query goes in the nucleus and rearranges the genome. So with these two levels of regulation, we have very tight control of the query, and then we can stabilize the genome without estradiol induction, or with estradiol induction, we can start scramble the genome and make the genome really flexible. So this slide shows you the effect of the scramble. So on the top is the wild type yeast BY4741 without query and with query induction. So this is the control. And in the bottom are the sensitive yeast. So without query induction, there's no difference between the sensitive yeast and the wild type. So this fits really well with the stable genome design principle. But once you add a little bit of query in nanomolar concentration, you start seeing scramble effect on the sensitive yeast, because then you start seeing about 100-fold killing rate. This makes a lot of sense because when two lox psi recombined, we cannot control which two lox psi recombine, and very often we delete essential gene. Once we delete essential gene, the cell die. So what's interesting here is what's left after the scramble. So there are about 1% of the yeast are survived. We purified these survivors and then we sequenced about 100 of them. From the deep sequencing data, we discovered these 100 genomes we recover after the scramble, they're all different, which is really encouraging because that means our system actually fits the flexibility design principle. We actually can diversify the genome by scramble. And even more encouragingly, we don't see any off-target recombination, meaning when there's a rearrangement, it always happens between two lox p sites. So I have been talking about scrambling. So we actually have a really cute acronym for it. So it's synthetic chromosome rearrangement and modification by lox p media evolution. So if you see the genome as a deck of cards, and each gene is a card in the deck, the scramble system gives you a way you can shuffle the deck of cards. You can rearrange the order of the genes. You can invert some of the cards in your deck. You can duplicate some cards. And this gives you infinite possibility of rearrangement of a genome. So the idea is after we finish the synthetic chromosome, synthetic yeast, as a homogeneous population, then you give it a shot of query, and every two lox p site can recombine. So in principle, you are going to generate billions of synthetic yeast genome. So we're not just building one synthetic yeast genome, we are building billions of them at the same time. And then you can screen for different phenotypes. So with the scramble system, we developed a new project called Induce Evolution of Sensitive Yeast Genome, EASY. So this is funded by Bio with partners in UK, France, Germany, and the USA. So the idea is we're going to take the sensitive yeast strains, put them into a chemostat. And in the chemostat, we can have different conditions, such as high pH, high temperature. And this is what I think is equivalent to a cage fight for the synthetic yeast. You put it in a condition which has high value for industrial setting, and then you turn on Cree. So all in a sudden, you have billions of synthetic yeast genomes in one chemostat. They're going to fight against each other to dominate the population, and the loser will be washed out. So every 10 days, we do single colony purification to fish out 20 winners. And then we put these 20 winners back to the cage, let them fire again. We just iterate the process 5 times. So this 50-day cycle, at the end of the 50 days, we're going to fish out 10 winners, and then we are going to do whole genome sequencing to figure out what makes a winner. We're not just going to look into the 2D DNA sequence, but we are also going to do 3C and 3D to do genome structure capture, because we think the chromatin structure might be also really important to make a yeast a winner in a particular environment. So with this powerful direct evolution approach, we will be able to direct the yeast genome towards desired phenotype. So I told you we made the first chromosome as a bag or yak. As a hit or miss approach, it's a bit dangerous. It's equivalent to take the operating system from your computer and do all kinds of crazy modification to it. And then you put back the operation system and try to reboot your computer and hope for the best. So this is not a very engineering-like approach. So we have come up with different ways we can synthesize the genome, but closely monitor the fitness. So what we do now is we synthesize 30,000 base pair sensitive DNA, and we will target with a marker Lu2, and then with a bit of homologous region on the right-hand side of the marker, and then we transfer these 30,000 base pair DNA into the genome, and it recombined with the Y-type chromosome, and they wipe out the Y-type DNA, and then we select for URA3. And in the next iteration, we synthesize the next 30,000 base pair, has homology region to the left-hand side of the first marker, URA3, and then we tag the DNA with another marker, Lu2, and homology region to the right-hand side with the Y-type chromosome. So you replace the next 30,000 base pair, and you select for URA minus, Lu plus cells. So with just 2 markers, you can systematically wipe out the entire chromosome and replace the Y-type DNA with sensitive DNA. So this is a much more engineering-like approach. So we can closely monitor what happened to the cell when you replace the Y-type DNA with sensitive DNA. If at some point the cell becomes really sick, or even you don't get any cell, then we will know it's very likely due to the last 30,000 base pair we just transform into the cell. And then we can go in and try to debug from that 30,000 base pair. So with this new approach, now we can break down the chromosome to large chunk, which is 30,000 base pair. And then we further divide the large chunk into chunk, which is 10 kb. And finally, we synthesize this as a building block. This is 750 base pair, and then we sequence everything with Sanger reads. So with this new approach, we recently just finished 770,000 base pair of chromosome 2, and this is in collaboration between my group in Edinburgh and Beijing Genomics Institute in Shenzhen. On the left-hand side, I'm showing you all the designs we have done to the chromosome SEN2. We remove a lot of repetitive elements, and we also delete about 13 tRNA genes. We insert 367 LOXP synthesize into this genome. On the left bottom it shows you a lot of PCR tagging we have done, and this has been very tedious, but we have been using the PCR tag to verify the sensitive DNA incorporation every time we transfer a piece of DNA into the cell. And the right-hand side shows you the design footprint of the chromosome 2. So as I told you, constructing the first 100 KB was outsourced by a company called Codon Device, but commercial synthesis is really costly. So we come up with the idea, can we harness the awesome power of undergraduate students? And we set up a class called Be a Genome in Johns Hopkins. We teach undergrad students how to synthesize DNA. And they love the course because they feel this is really a fusion between a biotech company and a graduate school. So when they first come in, they have an interview with us. Once they pass the interview, they were given a key so they can come to the lab 24/7 to synthesize DNA if they like. Every week, the students need to come up to present their data, and they need to dissect the failure and come up with the next experiment they want to do. So this gives them a flavor of being in graduate school. And also, they were assigned with milestone to synthesize 10,000 base pair every semester. So this is more like a biotech company. The students really love the course, and we have really good course feedback. Last year, we successfully translated this class to China. So we set up a sister class in Tianjin University to teach undergrad students how to synthesize chromosomes. So the student has been doing great. They synthesized the world's first sensitive chromosome, chromosome 3, in 12 months. And this is a paper we published last year in Science. So this quickly became an international effort to finish the sensitive yeast genome. So now we have almost 10 universities participating in the consortium across 4 continents. So this slide summarizes what we have been doing. So we designed DNA sequence in silico. We transmit the sequence digitally by email to different sites. And then we in vitro synthesize the DNA and in vivo assemble the DNA into a sensitive yeast. So when I moved to Edinburgh, I started realizing automation would be the future for sensitive genome engineering, because automation is pervasive in industrial for a couple of reasons. It's very precise, it's consistent, and you can work 24/7, and also it reduces labor costs. And I really see DNA synthesis and assembly not a biological question, as more manufacturing problem. Each step of the DNA synthesis and assembly are not difficult to automate. But each step you have failures, so we need to reschedule the failures, and it quickly becomes a process engineering problem. And some protocols are better than the others for automation. For instance, if you can do one-step incubation, that's much better than running a PCR and gel extracted. So with this in mind, we are going to launch what we call Edinburgh Genome Foundry. So this is the national facility in the UK funded by BBSRC to set up a fully automated genome synthesis center in Edinburgh. So I'm showing you the design of the space. So this will be finished by the end of this year. If you come to Edinburgh, you will see the robots moving around in the glass room. And this is the setup of our Genome Foundry. As I can point out here, is we have 2 acoustic dispensers, and these are the key to our foundry operation. As I already mentioned, acoustic dispenser are leaf handler machines which use sound wave to manipulate nanoliter scale liquid. So in contrast to the traditional leaf handler which use tips to dispense microliter, we can use the echo machine to dispense nanoliter liquid with sound wave. And this saves a lot of money on reagent, and also it doesn't use any conductive tips. So we save a lot on tips and avoid the cross-contamination. So we use echo to successfully downscale PCR from microliter to nanoliter. We were able to get PCR working in 250 nanoliter in our hands. And this has been great because we run a lot of PCR tagging for genome typing. With the echo acoustic dispenser, we also be able to downscale the popular Gibson assembly from microliter to nanoliter. So what we're showing here is we were able to downscale the assembly of a plasmid from 20 microliter to 500 nanoliter. So effectively, we cut the reagent cost by 40-fold. But it's not just saving money, but also the assembly efficiency. So if you look at the right bottom figure, that shows you the assembly efficiency. Actually, we achieve better efficiency in assembly and also save a lot of reagent cost. Similarly, we also be able to downscale golden gate reaction from normally 7.5 microliter all the way to 50 nanoliter. So this saves even more on reagent, but the efficiency is actually much better than do it by hand. So the fact of being able to downscale DNA assembly from microliter to nanoliter really improve our workflow efficiency. So again, I'm showing you the PCR tag in our project. So remember, we need to run a lot of PCR tag across the entire genome. So this is one example of chromosome 2. So for this chromosome alone, we need to run about 1,000 PCR reactions. So the student need to set up the reaction by hand, run a PCR, and load it into a jail. And this is really tedious, and it's really also very costly. So with the echo acoustic dispenser and those qPCR machines we have, we were able to replace the manual PCR tagging with qPCR tag, which we use the echo to set up nanoliter qPCR reaction, and then we run endpoint qPCR in a light cycler. With the output, it can give us a digital output of where a piece of DNA is sensitive or wild type. So with our collaborators, Jay Booker and Nathan Mitchell at New York University, we successfully set up our qPCR tagging here in Edinburgh, so we can run 1,536 qPCRs in 15 minutes with our echo setup, and also downscale our reagent cost to about 7 cents per reaction. And this really speed up our progress in making the sensitive chromosomes. So with that, I would like to close by thanking the organizers of this webinar, and I'm happy to take any questions you may have. Thank you very much.
Thank you, Patrick, and thank you, Randy, for your outstanding presentations. You've both done a great job in showing how acoustic liquid handling technology can advance synthetic biology in a number of areas. And our audience seems to agree based on the questions we have received, so why don't we take the first one?
All right, everybody. Time for some questions. So our first question is going to be for Randy. Randy, question one of our audience members would like to know is, can acoustic energy from the echo damage samples?
No. The acoustic energy required to transfer a sample is relatively low. No heat is generated or anything like that. Many echo users regularly transfer chemical and biological samples every day without any issues of any kind.
Okay, thank you very much, Randy. Our next question is for Patrick. Patrick, audience member asks, it's becoming increasingly obvious that chromatin is not simply a DNA sequence. How will you measure effects on three-dimensional chromatin nuclear structure?
That's a really good question. So we know, DNA sequence is not just the only cause which can define the life. The chromatin structure really play a big role in defining the function of the genome. So there are technology called 3C and 3D chromatin structure capture. So you can use that technology to visualize how the chromatin was packed. So that's the technology we use in sensitivities to observe, how the chromatin structure influence the function of the genome. And we are collaborating with groups in Pasteur Institute to look into the chromatin structure before and after scramble. So we are really interested in how to use that information to influence our next iteration of design.
All right, thank you very much for that, Patrick. Our next question is for Randy. Randy, are there any issues with evaporation in miniaturized reactions?
That's a good question. Evaporation happens. It can be an issue with certain solutions. However, with plate sealing and use of our microclimate lids, we've been able to avoid any problems. Typically, just unsealing the plate or delidding the plate before it goes into the ecosystem and then sealing it right after solves the problem.
All right, thank you, Randy. Patrick, our audience member, asks, what about effects on splicing of the few genes that contain introns?
So in our original design, we take all the introns off because we don't think alternative splicing is essential. But to be honest, there are not many introns in the yeast genome to start with, so we only got just over 200 introns in the entire genome. We get 6,000 genes in total, so it's every out of 30 genes has an intron, so it's not many of them. But later in the design, we see publication came out talking about ribosomal intron might be quite important for the fitness of the yeast. So we revised our design. So we put all ribosomal intron intact. So we didn't remove all these introns. So this is a way we can quickly change our design strategy to reflect the latest scientific finding. And as a real agile way, we can quickly revise the design and remake the DNA and rescue a viable yeast.
All right, thank you, Patrick. And it looks like we have one more question for you. Our audience member asks, how will you be able to assess non-coding functions of DNA sequence?
So we didn't change all the, we didn't move the non-functional elements, but instead we put in the LOXP site. So we let the LOXP randomly recombine with each other and randomly lose, these elements. And then using the LOXP scramble system, we will be able to see what combination would lead to a viable product and what would not lead to a viable product.
All righty, thank you very much. And it looks like with that, we are at the end of our webinar. So I'd like to remind everyone that the webinar will be archived on our site for 6 months at www.genengnews.com. So if you missed parts of it or want to watch it again, feel free to forward the link to your friends and colleagues as well. I'd like to thank Patrick and Randy again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to LabSite for sponsoring this webinar. So hopefully we'll see you again soon at another Gen webinar in the near future. Goodbye for now.